Now I'd like to welcome someone that you all know. We're thrilled to have with us Emmy-winning award winner, Chief Medical Correspondent for CNN, and practicing neurosurgeon, Dr. Sanjay Gupta.
Thank you very much. Thank you. I'm really honored to be here. What a day. Really remarkable day. I realize, by the way, it's about 8:30 on the East Coast, so good morning to many of you out there in the audience. I just want to take a moment before I start to just revel for a second how extraordinary this all is. I mean, we got an audience with the Pope, an audience with Vice President Biden. We're sitting here talking about stem cells and cell-based therapies. The Vatican. It's a really remarkable thing, and I just want to take a second and congratulate the organizers of the conference for all this. I've never been to a conference quite like this, and I'm learning a lot, but just also really reflecting on how monumental I think this is all going to be. One of my favorite quotes from one of my professors in medical school was that most of science has a faith-based component to it. And I've thought a lot about that as I was preparing for this conference and preparing for these panels. There is a faith-based component in a lot of what we do, whether it's in the lab or taking care of patients. I'm a neurosurgeon. I take care of a lot of patients with brain tumors, and there's a faith-based component in what I do. When my mom got cancer, I did everything I could for my mom. You know, who wouldn't, right? And as a doctor, certainly you have lots of skills, but in the end, there was a lot of prayer and a lot of faith on behalf of my mother as well. As much as we focus on the science, I think we focused a lot in this conference on the stories, and we do that as to really make the science stick. As journalists, we do this because we want to tell stories and make sure that the lessons that we're getting across on television are things that are going to be memorable in some way, things that you're going to want to share with your family and your friends, and that's a lot of what we're going to do today as well. I'm going to spend a lot of time making sure that we define terms over the next couple of panels. So if I look out and I see some confused faces, I may try and take a step back and define some of the terms that we're talking about. I also want to make sure that things are really crystal clear. There's a lot here. There's a lot that's being discussed. Some of it is complicated, so I'm going to try and make it as clear as possible, such as the topic of our next panel, an important topic, and I think I'd start off by saying this. The treatments that we're talking about, as wonderful as they are, oftentimes cost too much. There was a recent study that came out that showed that the United States cost of cancer will jump by 27% in the next 10 years. It's $158 billion annually by 2020. We've heard a lot about surging drug prices. That's been a big topic in the news lately, and the impact on cancer patients. You saw some of the slides earlier, more than $10,000 a month for some of these medications, and obviously that's not acceptable, but it's only part of the problem as well. So how do we reduce some of these inefficiencies in drug discovery? What does it mean for all of us as potentially people who treat these cancers, potentially people who are affected by these cancers? How do we develop therapies quickly that we know are actually going to work on patients' disease, that sort of gap between drug discovery and impact? And how are the providers and insurance companies going to play an active role in all this? How are they going to be part of that solution? So to discuss all of this, and I think there's a lot here, but I think we're going to get to it, but discuss all of it, please join me in welcoming Dr. Andrew Pecora. He's founder and executive chairman of COTA in New York. Steve Kafka. He's an economist. He's the president and CEO of Foundation Medicine in Cambridge, Massachusetts. And Dr. Glenn Pomerantz, chief medical officer of Horizon Blue Cross Blue Shield. Welcome, gentlemen. I guess I'll just start off by asking you all the same question. I know you have presentations, and then we'll have some questions and answers after that. But how are you using data to improve and personalize healthcare? Dr. Pecora, I'll start with you.
Yeah, so in the simplest of terms is making sure every patient gets the right care they need, no more and no less, through data.
Yeah, Sanjay, our objective at Foundation Medicine is really to understand each patient's individual unique cancer at the level of its molecular blueprint. We know that cancer is a disease of the genome, as was mentioned earlier by Carl June today. and really understanding each patient's unique cancer and then bringing that information back into the hands of physicians and to biopharmaceutical companies as they develop new medicines to really help drive better decision-making in both direct care decisions as well as drug development.
Let me augment Andrew's comments. For us, the key is the variation and the unwarranted or ineffective care. So we can get precise data and get the right treatments matched correctly, then that's where we need to be.
Okay. And Dr. Pecora, I know you have a presentation, so why don't you go ahead and do that, and then we'll ask some more questions at the end of this.
Great, thank you very much.
Sure.
So we're going to change gears a little bit. For those of you who know me, I'm a bone marrow transplant doctor. I presented here 3 years ago on my work in adult stem cells for cardiovascular disease. But as exciting as everything we've heard about so far, there's another major problem facing not just the United States, but all developed nations, and that's accessibility because of affordability. And my colleague Steve Glenn and I are going to tell you about a path we've taken and working together in a unique partnership to address this problem just as seriously as you've heard people addressing the problems of biology, and we're going to walk you through it. It's a bit of a story, but I promise we'll get you there. So, healthcare is in crisis. We heard from Vice President Biden the incidence of cancer is going to go up significantly, and that you heard yesterday and today these therapies are incredibly expensive, $150,000, $200,000. And there are countries that are now trying to take approaches to address this. Certain countries are doing prospective assessment of value where they decide, is this really worth it to pay for our citizens, considering what percentage of the gross domestic product healthcare is costing us. And other countries are trying other novel strategies to address this by changing reimbursement. But none of these ideally line up with the goals of precision medicine. So we really need to decide, as citizens of our countries, are we creating these new treatments for the privileged few, or do we want everyone to have access to them? And if so, what can we do about it? So there's a new term in the United States. As an oncologist, we talk about toxicity. Do you have nausea, vomiting, hair loss, and things of that sort? But there's a new term. It's called financial toxicity. 62% of bankruptcies in the United States are due to healthcare costs. It's the #1 cause of bankruptcy now in the US. And it's not just a problem of indigent and uninsured patients. Most are well-educated, homeowners, middle class. 75% of these people had health insurance. The percent of bankruptcies attributable to healthcare costs rose 50% between 2001 and 2007. Is this just limited to people who are going bankrupt? At COTA, which is a big data precision healthcare analytic company that I created with my colleagues at Hackensack University Medical Center and we spun out, we started to look at these things in a serious way. COTA created a score called Living with Cancer. It's a questionnaire that asks all sorts of questions about toxicities, lifestyle issues, but also finances. And what we found in a sample of 941 random patients with advanced malignancies, that 38% of these patients reported moderate to severe financial distress. At a median income of $51,000, 48% expressed concerns. And what was really fascinating is the younger you were, the more you cared about it. And the reason most stated was, how will I support my family if I spend all this money on my health care? So the question is this hopeless? Are we destined to ration care? A colleague of ours, Don Berwick, who most people know, former administrator of CMS and a board member of COTA, has helped us see the path forward. The United States gross domestic product is $17 trillion. $2.9 trillion of that is expended on healthcare, 70% of our GDP. It's the greatest percentage of any nation, developed nation in the world, on GDP. So whether or not we should be spending that much, that's not the question. Look at the next circle. $87 billion of healthcare expenditures are considered wasted. That's 5.1% of the gross domestic product of the United States. So we made the decision, instead of focusing on the $2.9 trillion, let's focus on the $87 billion and see, can we do something about it? Because if we could, then that number of 17% would go down to 11.9%, and I very much doubt we'd be talking about a healthcare crisis, affordability crisis. So we at COTA turned this into a math problem. So you can't get your head around $87 billion, and by the time it adds up to that, it's too late. And this is not unique to the United States, by the way. I'm just giving you US numbers. This applies to many developed nations. It's just not at this scale. So it turns out it's $238 million a day, and that's 120,000 adverse variance a day, variance events per day. And what's a variance event? A variance event is you should have done something else. You did something that you should not have done, or there was something better that could have been done for that patient. So the question next is, well, how do you address these vari events? How do you find them? And the issue we came to realize, my colleagues at the John Thor Cancer Center and I, and colleagues from across the United States that worked on this, is that to get to true precision medicine, it's not just the disease. There's a person that has the disease, and that person has a family history, has behavioral patterns, certain age, certain weight. They live in a certain socioeconomic status. Those things matter. And you have to combine that with all of the issues about the disease. So you have to put those two things together. And once you start putting those things together, you instantly realize you can't do this in words. You could never do it. So what the sort of seminal moment is, we realized we had to digitize the biologic narrative. We had to turn it into numbers. Because zeros and ones, you can do a whole lot with. And what you're looking at is what's called the COTA nodal address, and this has been patented now by the US Patent Office. And let me walk you through it. So 01 stands for cancer. 02 would be cardiology, 03 dentistry, 10 is mental health, and 20 is urology. So all of medicine is being digitized. The next number, because 01 is cancer, is breast cancer. The next number is, it's a woman. A 1 is a woman, a 2 would be a man. I'll get to the phenotype in a second. The 1 is, what is the intent of the therapy? In cancer, is it adjuvant, meaning you're giving therapy after surgery? Is it neoadjuvant, before surgery? Is it metastatic disease that has been resected? Is it metastatic disease that's still there? And the last number is it your first presentation, second, third, and fourth? The phenotype is the embodiment of everything that's relevant about the person and everything that's relevant about their disease. So, well, that could be trillions of numbers. And if you actually take all of the published literature and that describes what's relevant, it's trillions of numbers. Well, that wouldn't work. So what we did was we spent about 3 or 4 years, my colleagues at Hackensack and colleagues across the country, and we looked at all of the published literature and said, what really matters? If you have this type of tumor, what else matters? And we were able to get it down to 570,000. So there are 570,000 and some small number potential COTA nodal addresses for cancer. 570,000 GPS points. But nature did us a big favor, because it turns out in reality, and this is real world data on the first 7,000, we have many more patients now, and it turns out in nature there's only several hundred. So even though there's 18,000 potential CNAs for breast cancer, only 700 exist, and that's because there's something called biologic disequilibrium. So if you have a tumor of a certain size, it's associated with other things and not other things. And that even compresses this further. And the CNAs aren't evenly distributed. And I'm sure this won't surprise some of you, the distribution of CNAs is directly tied to zip codes. So in certain zip codes, you have more aggressive advanced tumors than other zip codes. So how do you get at the problem? The goal is eliminate behavioral variance at scale, identify and eliminate it so that you can work on continuation of elucidation of biologic variance. And here's the formula we've been working on, and I'll let Glenn talk about this a little bit more later in payment reform, but it's called enablement of the Goldilocks formula. For every single patient, with precision, give them just the care they need, no more, no less. Now, you think you might know that, but actually most times you don't know that. So what we've started to do is we've started to assign CNAs to patients and then put them in what's called a bundle. So in this case, this is metastatic lung cancer. It could be a chemotherapy bundle. It could be an immunotherapy bundle. It could be a targeted therapy bundle. But there's not one immunotherapy. There's not one targeted therapy. There's not one type of chemotherapy. So you actually measure over time the yellow versus orange versus green versus blue. They all have metastatic lung cancer, but they have other attributes that make them different. And you see, if I'm a yellow CNA, which regimen is best for me over time? Where do I have the least cost and the greatest clinical outcome and the least toxicity? And you do this iteratively, and you could start to change reimbursement policy around this. And this is what we've started to do. So does variance really exist? This is real-world data. And you can see for CNA 0111, DSC means disease-specific concentrated doctors. You don't want to call them experts because you insult the other guys. So these are academic, well-known medical centers. And non-disease-specific are community practicing oncologists. And you can see for that type of CNA, they did exactly the same thing. Everybody got hormones. You get a little bit more advanced breast cancer, and you can see it's very different. And in fact, the academic doctors gave more chemotherapy than did the community doctors. Which one was right? We don't know. We're going to find out. Another real-world data set. This is Alcon EGFR testing. This is mutations that if you have them in lung cancer, you can get a targeted therapy. Everyone should have these measures. These are well-known centers. Look at the statistics. 49%, 52%, and 75% testing rate. It should be 100%. Why isn't it 100%? We're now starting to study that. What's the consequence of not getting tested? If you have an EGFR or ALK mutation and you get a targeted therapy, your median survival is 44 months. If you don't have one of those mutations, your median survival is 11 months. You live 1/4 of the time. Imagine the 30% of the people who weren't tested who could have gotten the targeted therapy. 1/3 of their life has been taken away from them. Unacceptable. This is my last slide. And so, can you really do more testing and find value? Can this really work at scale? And so what we did is we looked at thousands of breast cancer patients, and we dove into the genome. There's, as you know, about 20,000 genes that code for proteins. There's about 1,000 of them or so that we think are related to cancer. We looked at 21 genes through a test called Oncotype DX. And for women with stage 2 breast cancer, what we found was if you did the test, which cost about $3,800 retail, that you saved $11,500 for every patient, regardless of the result of the test. So if I were king, I would make sure that every woman in the world would have this test who had stage 2 breast cancer, because you would save money by avoiding unnecessary chemotherapy. And that will allow you to afford to do the test, which actually cost an incremental $4,500 for stage 1 breast cancer, where a small percentage of patients benefit from chemotherapy. So this is just the tip of the iceberg. Now we're going to dive even deeper into the genome to show you how we can find even greater value.
Great, thanks, Andrew. I am really excited not only to be here, but to be working with these gentlemen in this novel partnership to really tackle these questions of access and affordability. And as I mentioned when Sanjay kicked us off here today, we're really committed at Foundation Medicine to delivering precision medicine today, and we're committed to delivering this today for patients just like Kristen, which I'll show you in the video just now.
I have colorectal cancer. We had to do a surgery. We had to do radiation to shrink this tumor. A lot of people were, oh, your cancer's not going to come back. Okay, positive thinking. But at the same point, you need to also be realistic. I was no evidence of disease and did the foundation testing there so that I would have some tools in my toolbox for if and when my cancer did come back. My cancer came back in June of that year, but I had the report where I could really read through it. These are the mutations you have. This is what your cancer is. And these possible things can work for you as an individual. I'm on 2 drugs that are for breast cancer, but I don't have breast cancer. No one would have thought, oh, let's try these 2 drugs unless I had the testing done by Foundation. They gave me 3 months, and it's been 4 years, and it's because of thinking outside the box. If Foundation Medicine didn't exist, I wouldn't be sitting here today.
That video was taken last fall, and I had the privilege of seeing Kristen just a couple of weeks ago, and I'm happy to say she continues to do well. Her story is really a powerful embodiment of a transformational new approach to precision medicine that, as I said earlier, starts with understanding each patient's cancer at the level of its molecular blueprint. It's been clear over the last day and a half already that we are living in the midst of a really unique and exciting moment in time when this explosion of cancer biology is colliding with rapid advancements in information technology. We have the confluence of many new molecular tests and new medicines and hundreds of companies studying thousands of different clinical trials. So very exciting, but against that backdrop of complexity, we are asking a lot of doctors and payers as well. Already they're overwhelmed with information and with the pressures of running a business, and we're asking them to make costly and patient-critical decisions where each patient's cancer is really as unique as a snowflake. So how do we solve this conundrum? Well, part of the answer is clearly in the COTA approach that Andrew has just described, and a critical part of the solution is understanding each patient's cancer individually and comprehensively. So let's remember that when doctors do tend to order molecular testing today, and which is not as often as it should be, as Andrew shared, they generally order one test at a time, maybe a small panel of genes, and they're looking for pre-specified mutations. So this approach consumes precious biopsy tissue, cost, and time, and it really limits the insight to a very narrow view of the cancer. Well, we have improved upon these limitations with what we call a comprehensive genomic profile. Our flagship tests really shine the floodlight on each patient's individual cancer. We're looking at more than 300 genes across the entire coding region of those genes. And to do this, we only need a small amount of biopsy tissue or blood. About half the size of a garden pea is all we need for our solid tumor test. So our tests are tissue sparing, saving doctors and the system cost and time. And using our approaches to capturing, sequencing, analyzing, and feeding that information back to physicians, we return results in less than 2 weeks in a format that's easy to use and simple to digest. So by shining the floodlight on each patient's individual cancer, we are really leaving no stone unturned in generating options. And just take Kristen's case from the video. The mutations that we found in her colorectal cancer that led to her treatment would not have been found by a standard testing approach. But this is, while it's simple to draw a floodlight on a slide, it is really not simple to do. And with critical patient care and healthcare dollar decisions at stake, we as a community, I would urge, really must demand that this is done well. Quality really must be at the very top of our priority list. We've followed that notion from the very beginning, and our tests approach 100% accuracy across all key measures, and we publish that data in leading peer-reviewed journals, and we're working with regulators and with payers to really set the benchmark for this kind of an approach. We do all this because it's critical that we deliver information that physicians and payers can trust, because as I said, this trusted data is really driving these enlightened treatment and payment decisions. But to make this information useful, we have to be more than a testing company. We also have to be a technology company. We are combining our tests with a database of now more than 70,000 comprehensive genomic profiles and are building a suite of technology tools that put insight into the hands of doctors and payers in a way that they can really use to improve outcomes and cost efficiencies. We've invested more than 9 million computing hours to analyze patient samples, and we currently have about 5 petabytes of data in our databases. That's more than 1 billion song downloads on iTunes, just to put it in perspective. But we have to take all that data and we have to make it simple and useful. So to do so, we've already created dozens of tools. One, for example, is something that we call our Smart Trials application. We've talked a bit about clinical trials here today. And what Smart Trials does is deliver back the information about the genomic profile and match that to available, relevant, and importantly, local clinical trials that physicians can point their patients towards. So this is really critical for addressing these goals of action, access, and affordability. Unfortunately, in cancer, with as much progress as we are making, information doesn't always lead to better outcomes. But this information is still a really important part of the solution because, number one, it empowers the patient to make critical end-of-life choices. But it also is important for the system because from a financial standpoint, it can take cost out where we no longer have interventional treatment options that are likely to work. And this is Cindy's story. I suspect many of you have stories that you know that are much like this one. Cindy was the 46-year-old mother of a 7-year-old boy. She was diagnosed about 1.5 years ago with a very aggressive form of breast cancer. She had surgery and adjuvant chemo, more additional chemo, and in the end, none of it helped. When we tested her cancer, unfortunately, we did not find any mutations that were linked to therapies or clinical trials on which her care team could take action. So upon receiving the results, her family decided to shift their focus to hospice so that she could focus on the comfort and quality in the weeks before she passed away, which was just last month. About a week later, we received a note from a family friend, and I want to share just a piece of it with you today, the email that we received. He says, "This experience didn't result in an outcome that helped Cindy find a new medicine, but the value you bring can't be measured by patient outcomes alone. The value that you bring to all the loved ones that are left behind is not measurable. They can go on with life without questions or regret, knowing that no stone was left unturned, that everything was done, and that this was meant to be." Cindy's case also teaches us that there remains a tremendous amount of work to be done. And we must continue to innovate, and we believe that information is the key to this. Earlier this year at President Obama's Precision Medicine Summit, we announced the release of an unprecedented database of genomic information on pediatric cancers. The scope and depth of this data doesn't exist anywhere else in the world, and we've now built a first-of-its-kind portal to provide free research access to the entire pediatric oncology community, leading to new insights that we believe can help lead to the development of important new medicines for kids. The sharing of this pediatric data is just one example of the ways in which we're working to break down silos and drive creative new solutions into the problems that we're tackling here today. Another example is what we're talking about doing together, the three of us, enabling payers to know what to pay for. The innovative partnership that we've formed with COTA, Horizon Blue Cross, and Foundation Medicine is testing this approach in non-small cell lung cancer, where we're using an upfront comprehensive genomic profile along with the COTA nodal address approach to measure changes in survival benefit and total cost when this information is used to guide treatment decisions. The effort builds on recently published literature looking at patients who had been tested, but tested negative in non-small cell lung cancer, when in fact what we found using a comprehensive approach, about 2/3 of them actually had genomic results directly linked to an approved therapy or a clinical trial. So that's a huge benefit for patients and for the systems, and we hope to demonstrate this first in lung cancer and then spread it more broadly. So the coming together of this panel today, I think, is really quite extraordinary. We're doing it, though, because it really holds the promise of achieving our shared goals of better access, better outcomes, and savings to the healthcare system. So together, we really believe that we are ushering in an age of molecular information in oncology, and that together, we are showing that information can really be a cure. Thanks.
Good afternoon. So 3 years ago, Andrew approached me. He led a group of over 100 oncologists in my state, and he said, "Glenn, we're going to digitize the biological narrative so we can measure value." And he said to me, and as the first proof of concept, I want you to meet Foundation Medicine. And so I went up to Cambridge and met Foundation Medicine, and they demonstrated to us the power of unlocking the human genome. And it became fairly self-evident very quickly that broader audiences, obviously you're the experts here in this room, that you understand that these technologies are fundamentally changing everything we do today in research, treatment, and payment. So whether you're a national payer like the National Health Service in England, or a single commercial payer like myself, you need to know what condition or disease was treated because that information will link to a payment schedule. We have a billing language, many of you are familiar with it, it's called ICD-10. It's used worldwide, and it translates diseases or conditions into numeric codes. It's based on an anatomical framework, not a molecular one. So let's take lung cancer, for example. There are codes to tell you which side of the lung it's on, which lobe, whether it's in the tissue or the bronchial tube. But what's more important is what it does not tell you. It does not tell you if it's local or metastatic. It does not tell you if there's a treatable driver mutation. It does not tell you if the patient's working or bedbound. As a payer, our billing language does not include essential information regarding outcomes and quality of care. So if you're going to pay for value, a combination of cost and quality, and you cannot define quality, doctors and hospitals correctly object that you cannot compare their panel, my panel of patients, to others based on your billing codes and billing information. ICD-10 is an obsolete classification system in the age of molecular medicine. It does not go deep enough to classify patients to understand outcome and quality. And it makes paying for value otherwise a futile exercise. Recently, I was in Washington, D.C., and I was giving this talk to U.S. corporate policymakers. And I was explaining Andrew's work. And I told them this story because they weren't familiar with ICD-10. And I said, remember the Swedish biologist, botanist, physician Carl Linnaeus, who in 1735 publishes his landmark System of Nature, a taxonomy that described the natural world around us. And we all learned this in grade school, and Sanjay, I know you remember this. Kingdom, phylum, class, order, family, genus, species. I know he knows it. So here's Carl Linnaeus. And what I want to do is I want to take you through an example. So imagine if Carl Linnaeus, instead of devising 7 levels, devises 5 levels of classification. Let's start with animal. And we want to compare one group for comparison purposes and look at different outcomes and treatments. Let's choose bears to go on the Goldilocks theme. Okay, so you can see you have a grizzly bear on the left, black bear, giant panda, red fox, all the way to a starfish. Animals are multicellular creatures that are capable of spontaneous and independent movement, fits all the criteria here. Let's go one level down. This group you can't compare, they're too dissimilar. Let's go to phylum Chordata. Those creatures with a backbone and neural tube, the starfish drops out, lives under the water. But see, too dissimilar, these creatures to compare and contrast. Let's go another level down. Class Mammalia. These are animals with mammary glands, hair, and an evolved neocortex or brain. So the coral snake drops out. We're still left with 3 bears, a red fox, and an Albert squirrel. Let's go one more level down, order Carnivora, flesh-eating, meat-eating animals. The squirrel basically subsists on pine bark and pine cones, so drops out. This is carnivore, but still we have 3 bears and the fox, very different creatures. We now go down one more level, the 5th level, family Ursidae, and we're left with the 3 bears. And this level is described as creatures that are 55 to 1,700 pounds, large claws, climb trees. The fox drops out. So we're done, right? We now can set up our comparator group with 3 bears. And the answer is, of course not. We can't do that. The grizzly bear and the black bear hibernate. The giant panda does not. The giant panda cannot hibernate because its diet consists almost 100% of bamboo. It's nutritionally so little in value, the panda has to eat continuously almost all day and average over 40 pounds of bamboo just to store enough energy to live. So it can't store enough energy to hibernate. These are very different animals and very different creatures. So this is the argument, just to finish the 7 levels here. So this is the argument oncologists and doctors are making to us as payers. You can't compare my panel of patients to someone else if my patient has more stage 4 patients in my panel. Metastatic stage 4 lung cancer is a 5-year survival of 1%. Stage 2A is 30%. How can we put those groups of patients in the same grouping? So the caption here, ICD-10 is basically a generic bear. It doesn't provide enough information. There isn't enough biological similarity here. So this was the talk I gave, and a very well-known US policymaker approached me after the talk and said, the 3 bears, now I get it. So that was that. So now Andrew just showed you what digitizing the biological narrative looks like. And you can see this solved a problem that has been plaguing us for decades. And that is to precisely group like patients together so we can measure value. And what doctors, oncologists, you in this audience, really what you were discussing and the objection of what payers were doing is that we were not being faithful to the scientific method. And specifically, we were not being faithful to the second tenet, pattern recognition. You cannot recognize patterns unless you group like things together to create the same biological identity. And that's what the nodal addresses do. They create the same biological identity. So in Andrew's talk, he showed how precise classification revealed that a large percentage of patients with non-small cell lung cancer were not being tested for driver mutations, Alcon EGFR. And the consequences, as the study showed, were very serious with those survival curves. Stage 4, median survival of 44 months, with non-mutated and not tested about a year. In Steve's talk, he showed how next-generation sequencing can reveal these mutations at an astonishingly accurate rate and save lives that we just did not save 5 years ago. So what I have here is how every payer, US, Europe, around the world, we look at diagnostic testing. And we look at it through three dimensions. First, we say, is the test analytically valid? Is it technically accurate? At Foundation, they're setting the standard in many ways that their NGS, next generation sequencing, is at 99.9% technical accuracy. The second is clinical validity. So the test is technically accurate. Does it tell you something about the disease or condition in question? So certainly we know that ALK is related to non-small cell lung cancer, so this would meet clinical validity if we were testing for that, for lung cancer validity. And finally, is there clinical utility? So it's technically accurate, it's related to the disease or condition in question. Will it impact treatment? Is there any chance that this could change treatment? So since there are driver mutations that are linked to targeted therapies, the answer is yes. So we pay for this as a single gene test or a small panel test looking for other driver mutations. Broad molecular panel testing, what Foundation Medicine does, is not covered by most payers, including my company. You're receiving information not related to the diagnosis about the other 300 some odd cancer genes, not the 7 you're looking for in non-small cell lung cancer if you're diagnosed with it. But the promise is that there might be another pathway that might be discovered if you sequence all the other genes and look at the relationships amongst those pathways and accelerate matching for cancer patients to targeted therapies. Additionally, Foundation Medicine made the argument, if you're going to sequence the 7 genes, it's not that much more costly to sequence all the known cancer genes, again, a little bit over 300. And then patients would know immediately, as new trials emerge, they will have the information in their hands to say, oh, I'm eligible for that trial. How would they know otherwise if they haven't been sequenced? So having precise data and understanding the speed of discovery that next-generation sequencing delivers, Foundation said, why don't you pay for our comprehensive molecular panel? So just a quick reminder, I mean, you're the experts in this room, but again, taking a small biopsy, lung cancer, you know, the specimen here in the middle is the size of a pencil lead when it's sectioned. It's about 1 millimeter by 10 millimeters, and then it's stained. It's the size of, you know, again, of a graphite core of a pencil lead. The next step, and this is one really that I want to show you, is that to do all 7 driver mutation tests, all these gene tests, you're almost going to generate 40 slides. And what those of you who maybe not work in lung cancer, what you might not know is a lot of times the lab runs out of tissue because there isn't enough taken at biopsy. So then what does the doctor do? What does the oncologist do, the pulmonologist do? Do they rebiopsy? Well, in the elderly, the complication rate can be as high as 20%. With, you know, a collapsed lung, then you need a chest tube, and, you know, some pretty serious adverse, you know, considerations to take in mind. So, oh, how do I go back?
Can we go back one slide, please? Yeah.
Thank you. So, in this case, we said there's enough evidence here, preliminarily, we should study this. Maybe we should change our approach and sequence and pay for comprehensive genomic profiling up front. So what we did is, my company, using COTA as the analytical precision engine to measure this, working with Foundation Medicine, we're going to pay for Foundation's test. And we're going to study comprehensive genomic profiling versus traditional lab testing, ordering, and procedures. And we're going to look at outcome and survival based on testing approach and total cost associated with each pathway. Having a precise, and I can't emphasize this enough, having a precise classification system gave us the confidence we could study these questions in partnership with leading scientists, oncologists, you in this room too, hopefully, and premier diagnostic and laboratory companies. Before I conclude, let me show you what digitizing the human genome can look like with metastatic breast cancer, and what molecular medicine can teach us when we analyze that data. And most importantly, when we create precise biological identity. Here's a screenshot from COTA's database for metastatic breast cancer. The point here is, look at the variation from initial diagnosis, first progression, second progression, third progression, and fourth. And for the most part, all these treatments are approved and paid for. And the take-home point here is, they all can't be right? They just can't be. Now, if we study these patterns, formulate hypotheses, make predictions, and test them, will help more patients. Sean Parker earlier today talked about the lack of standardization with big data. I don't know if he said garbage or rubbish, but I'll say rubbish. He said rubbish in, rubbish out. Well, this approach is a solution to that problem. Now the three of us, as mentioned earlier, and really, I mean, Steve really highlighted that this collaboration would not have happened 3 or 4 years ago. It just would not have. The three of us are working together as one team. We're problem solving together with one goal in mind, and that's to improve cancer survival in our lifetime by increasing access to effective treatment, match the patient's molecular profile. We're working, and this is what's inspiring, at the DNA nucleotide level. And it's wonderful to think that our children may view cancer as we did infectious disease in the last half of the 20th century. Not a disease to live in fear of, as they did in the first half of the 20th century, but a disease or condition readily treated and perhaps cured by a pill or a vaccine. I won't read the words on the slide, I'll let you do that, but they express the hopes of all cancer patients, and it's our responsibility to make it happen. Thank you.
Great, thank you. We just have a few minutes, so I'm going to try and get to as many questions as I can. So let me just start with you, Dr. Pecora, again. How much of this is about basically trying to eliminate unnecessary treatments versus finding necessary treatments?
It's both. It's actually both. So COTA is being used as a tool for discovery for the pharmaceutical industry already. They're looking at super responders and under responders to a given disease and then figuring out why. And the other is what you said, is to identify behavioral variance where there is no value. Actually, you potentially are even doing harm and spending money you shouldn't, so the money can accrue to the benefit of the future.
Steven, when you talk about getting all this data now, actually mapping one's genome and finding mutations that maybe you wouldn't have even known to look for. Sounds like you were describing in Kristen's case, for example. She was taking 2 drugs for breast cancer, but she had colon cancer.
Right.
You found these breast cancer mutations in her colon cancer. We're treating her for that. That's a great story, a great point. But what is the sort of, if you will, the signal to noise ratio? How do you know what is valuable in terms of the mutations you're finding? And are there, I mean, you may find mutations for which there are no treatments, right?
So in Kristen's case, I mean, interestingly, colon cancer is a disease area where it's traditionally been thought this was not a genomically driven disease, but in fact, there's more and more evidence that it is associated with genomic drivers, like in Kristen's case. You know, what we find, Sanjay, is that more than 4 times out of 5, when we test a patient, we return results to the physician that are potentially actionable. So that's more than 80% of the time. Now, there is a gap to fill, to be sure, between, you know, potentially actionable and can that physician actually get access to the therapy, whether it's a, you know, approved therapy or a clinical trial. And that's a big part of what we're working on with our, you know, big data information-based solutions too, is to help facilitate the whole process all the way to treatment.
So I guess, Dr. Pecora, you're sort of on the technology side, and Steve, you're sort of on the testing side. How does it work? What does it feel like for a patient? They go in, they have a biopsy. What happens next? Where does all this data get stored? Who has access to it? How is it used?
Yeah, so currently the data resides within the database of COTA and the healthcare system. In our case, it's Epic at Hackensack. The goal is to push it forward to the patient. As we learned, our goal is to see that every patient gets a CNA assigned, so they know, hey, everyone else who has my CNA across the country, when they went down treatment regimen 1, they did a lot better than treatment regimen 2, so now I have eyes on what I should do next. And in follow-up to the question you asked Steve, you know, there was an example where we have patients who get 7, 8 lines of therapy, none of which work. We're spending a fortune. They're in the hospital, in the emergency room. Then you look for a targetable mutation, you find it, and they're home, back to their families, much less expensive. There are certain biases that have been introduced to the narrative that expensive drugs bad. That's not necessarily true. You have to look at the cost against the total cost of care, and that's what we're doing.
Was this an easy argument for them to make? I know you're all in this together, but
Right.
At some point you had a patient like Kristen who they're saying, hey, we need you to approve 2 breast cancer drugs, even though she's got colon cancer. We want you to pay for these drugs. How do you get the providers engaged in that conversation? I mean, this sounds like a rather unique sort of partnership.
I think it's all about the data. And when you start showing, these early graphs and survival curves and testing data that's coming out of the early results, it's compelling. So when we saw it, we were floored. And we said, oh my goodness, are we not being perhaps progressive enough with medical policy? Do we have to change the way we think about it? So let's do it in a scientific way. Let's be faithful to the scientific method and do it together. And look at all the, the clinical outcomes, the most effective cost, pathways, and then publish. What's
Interesting about this panel is that we're not really talking about drug discovery. We're not talking about new immunotherapy or stem cells, any of those things that much of this conference has been discussing. You're talking about using existing therapeutics and making sure you're matching those up with the right patients. What would you tell somebody who's recently been diagnosed with cancer? What should they do that maybe they're not doing or they're not getting right now?
Well, first of all, it's about following the evidence and about the accepted, no question, the approach in terms of either testing or treatment. If you want to use NCCN as a standard example. But beyond that, we have to allow, and this is what was so insightful for me when Andrew approached me, is that payers, we can't restrict doctors from being innovators on the front lines. We can't be so prescriptive we're following a pathway where we don't know what the right answer is. So we have to build a payment model around you that gives you that freedom to innovate on the front line or in world-renowned research centers, which many of you run. So for us in the United States, we're looking at this more as a bundle approach with the right quality metrics to support the innovation. So it's completely changed our thinking around payment that we previously used to deploy.
And we're out of time, but again, just to be really specific, Steve, would you recommend anyone who's diagnosed with cancer get their genome, their exome sequenced as part of their overall diagnosis?
I think, first of all, information is power for sure, and we gave a couple examples of that, whether it's leading to treatments or whether it's not. I do think that there, today is a set of patients that are with later stage disease, beyond sort of your standard surgery and radiation and standards of care therapy, where yes, it's absolutely appropriate to get this information. And I might just add really quickly that there's a really important link here too to I think our discussion this morning about immunotherapies, because from a cost, it's very exciting what's happening obviously, but from a cost expensive perspective, we saw how expensive those are. And we are learning a lot about what drives response to immunotherapies, including things we may be able to learn directly from genomic profiling. For example, patients who have a very high mutation burden in their tumors, hypermutated, are associated now in lung cancer and in other cancers with differential response to those PD-1, PD-L1 inhibitors. And so there's something in what's happening with that hypermutation and we see that in our tests and can provide that information on a research basis to docs to help them understand which patients are going to respond. So I think we're at the tip of the iceberg here and a lot of exciting things in front of us.
All right, gentlemen, thank you. Please give them a warm round of applause. Thank you.